So I decided to do a preview of DNA replication because tomorrow I'll be posting a video of DNA replication in a more intricate way, like a more complicated, more lengthy video. But I wanted to give you a kind of sneak peek or more preview of DNA replication. And so I'm going to be talking about terminologies, enzymes, DNA polymerases, fragments, short sections of nucleotides in this video. And all the on how all of this makes DNA replication function. And this will be a short video because it's just a preview. So here, topoisomerase. Okay, what it does, it unwinds the DNA, or essentially it relieves the stress of supercoiling on the helix, right? So you can imagine as the replication bubble is getting smaller and smaller, you know, as the process continues, stress is being built up because the helix is getting shorter. And so as the helix is getting shorter because the replication bubble is getting shorter, the twists or the turns get smaller and more compact. So this will build a lot of stress. So the isomerase, the topoisomerase, comes in and it unwinds the DNA and it relieves all the stress. And then now we have the helicase. Helicases are essentially enzymes that break the hydrogen bonds, in order to get the process, prepared for DNA replication. So it quote unquote unzips the DNA. Single strand binding proteins um prevents the two strands, the leading and lagging strands, from joining back together or annealing. The DNA primase sets down an RNA primer, right? And this is important because it initiates DNA polymerase activity, right? And the reason why it initiates DNA polymerase activity is because the RNA primer has a 3' hydroxyl group attached to it. So this helps DNA polymerase start synthesizing for nucleotides, right? As we've already explained right now, DNA polymerase obviously synthesizes. So we've already covered DNA polymerase 3, which is the main synthesizer of this whole process. And so what we have here is now DNAligase, which is essentially basically this enzyme which joins phosphodiester bonds between Okazaki fragments and DNA polymerase 1. And DNA polymerase 1 basically removes the RNA primers set down because the RNA primers cannot be replicated in DNA replication, right? Because they're RNA nucleotides. So the DNA polymerase 1, however, helps replace them with DNA nucleotides in order for as much genetic material to be replicated as possible. And so don't worry about this poly alpha. I just wanted to be talking about DNA polymerase 1 and DNA polymerase 3. I know they're not really on the diagram here, but DNA polymerase 1, remember, it removes the RNA primers and replaces it with DNA nucleotides, and DNA polymerase 3 is just the main synthesizer of the strand. And so, like I said, DNA ligase forms phosphodiester bonds between Okazaki fragments and DNA polymerase 1. And so a phosphodiester bond is kind of this covalent bond that joins bonds between base pairs on one strand, right? And so Okazaki fragments, you're wondering what do they do? Well, they make the lagging strand discontinuous, right? And so the lagging strand is read from 3' to the 5' end, right? And then, however, the leading strand is read from the 5' to the 3' end. And we actually have covered kind of the quote unquote sneak peek of this whole process. I mean, this is obviously not as intricate as it can be, but like I said, I'm going to be posting another video tomorrow talking about DNA replication in a more intricate way, right? In a more explained and lengthened way. And so I wanted to do this video so I can give you terminologies. That way, when you come in tomorrow and you watch our video, you know the terminologies and you won't be confused, and we can just run through them. And yeah, I think that was a very good sneak peek. And if you kind of like this video, I want you to subscribe, share this video with your friends if you think they will be interested, drop a like, and yeah, just stay tuned for more videos. General understanding, and thank you for watching this video. Yeah, so stay tuned. Thank you.